Welcome to Carbo Fitness, your best fitness channel. Ever wonder where do we get the energy to perform simple or even difficult tasks throughout the day? For example, simply walking or sprinting. But now, today's episode of Carbo Fitness, we'll be talking about how do carbohydrates actually give us energy to perform these simple or even difficult tasks. We will see how carbohydrates can also be used as different functions, not only for energy. Find out soon on Carbo Fitness TV. I'm Joan Kokkalis.
And I'm Mathew Marques.
And we are Carbo Fitness.
In this episode, sports meets science to get you performing at your 110%. With this knowledge, you will never lose energy again and always be one step ahead of your opponents. You might have heard of carbs, steroids, lipids, and proteins before, but you have no clue what these terms mean. Now we'll show you how understanding what carbs and their function is the key to your fitness success.
Mathew, what are carbs?
Well, John, carbohydrates is a macromolecule also known as saccharides, which is just a Greek word meaning sugars. Now, the basic structure of carbohydrates. They're made of molecules that consist of a carbonyl group and several hydrogen and hydroxyl groups, bonded on either side of a carbon. Therefore, the main three elements making up a carbohydrate group are oxygen, hydrogen, and carbon. Carbohydrates can be seen in two structures as they exist either as a straight chain or ring form. The ring is more common since it is more stable.
Fitness would like to present Professor Marques from Yale University.
Now that we described the basic structure of a carbohydrate, I will illustrate the differences between the straight chain and ring forms. As you can see here, there's a clear distinction between the two forms. The straight chain is merely 6 carbons bonded together in a line with substituents on either side. Okay, so now over here, the ring form is 6 carbons connected in a hexagon, while one of them is connected out of the ring.
Thanks, Professor Marques. Back to Carbo Fitness. Carbohydrates can be simple sugar or complex molecule that contain multiple sugar and can be separated into 3 different groups: monosaccharides, disaccharides, and polysaccharides. Monosaccharide is a simple sugar since it is a monomer that composes of one carbohydrate. A monomer is a small molecule that can covalently bind to other identical molecules to form a larger molecule. Disaccharides are just 2 monosaccharides joined together by covalently bond. Polysaccharides are many monosaccharides bonded together that form the long chain as you see here. Back with Professor Marques.
Now that we described the 3 different groups of carbohydrates, we can now see how they actually look like through this illustration. Over here, we can see there's a clear difference between them, as this is a single monomer we called a monosaccharide. This would be the disaccharide as it connects 2 carbohydrates, and this long chain would be a polysaccharide.
Thanks, Professor Marques. Back to Carbo Fitness. Now that we know the general structure of a carbohydrate, we can start looking at how to assign the appropriate numbers to the carbons inside the monosaccharide ring that we see earlier on. Back with Professor Marques.
By looking at this monosaccharide ring, we can start numbering the 6 carbons making up the carbohydrate ring. First, we look at the oxygen in the ring here, and start numbering the carbons clockwise from this oxygen. The clockwise would be like this. So let's say you had a clock, and you start going to the right, like that. Therefore, the first carbon would be here, second carbon would be this, third, fourth, 5th, and finally the carbon that is not in the ring but rather attached to it will be the 6th carbon over here.
Interesting. Thanks, Professor Marques. Back to Carbo Fitness. A straight carbohydrate chain is an open chain of carbons with a carbonyl group located on the first carbon in the chain and with either a hydrogen group or a hydroxyl group bonded on either side of the carbons. As seen earlier on, there is a clear distinction between the carbohydrate ring.
This carbohydrate ring can be seen as either alpha glucose or a beta glucose, which are isomers of each other. Therefore, the only difference between these two isomers is the positioning of the hydroxyl group. The position of the hydroxyl is dependent on the location of the aldehyde when the ring forms. On the alpha glucose, the hydroxyl group is always trans, meaning opposite to the 6th carbon in the ring. Beta glucose, on the other hand, always has its hydroxyl group attached to the 1st carbon, cis, meaning the same side to the 6th carbon.
With Professor Marques.
This carbohydrate ring can be seen either as a alpha glucose or a beta glucose, which are isomers of each other. Therefore, the only difference between these two isomers is the positioning of the hydroxyl group. Over here it's below, and over here it's above. The position of the hydroxyl group is dependent on the location of the aldehyde when the ring forms. On the alpha glucose, the hydroxyl group is always trans, meaning opposite, to the 6th carbon in the ring, which over here is the 6th carbon, therefore it'd be trans, meaning opposite. Beta glucose, on the other hand, always has its hydroxyl group attached to the 1st carbon, cis, over here, meaning the same side to the 6th carbon, which is the same side as the 6th carbon over here. Same side.
So this is trans and this is cis.
Meaning the same side, opposite side.
Interesting.
Alpha, beta.
Thanks, Professor Marques. Back to Carbo Fitness with the rest of the show. Now that we know more details about these carbohydrate rings, we'll be looking at how monosaccharides bond together to form polysaccharides. This is done by a condensation reaction to form glycosidic linkages. A condensation reaction, aka a dehydration reaction, is a chemical reaction in which carbohydrates are covalently bonded through the removal of a hydroxyl group from one molecule and a hydrogen group from the other molecule to form water.
However, there are two different types of glycosidic linkages, alpha and beta, in which contrast to the orientation of the linkages. The alpha linkages are trans, meaning opposite to the 6th carbon. The beta linkages from the 1st carbon to the oxygen is always cis, meaning the same side as the 6th carbon. Fitness would like to welcome Professor Kokkalis from Illinois Institute of Technology. Welcome, Kokkalis.
Thank you. I would like to explain the differences between an alpha glycosidic linkage and a beta glycosidic linkage. As we see here, alpha glucose will be bonded together through a condensation reaction that produces the removal of H2O from a hydroxyl group of one of the rings and a hydrogen group from the other ring. This will cause the formation of H2O outside this ring, cause right here. This, as you can see here, as an alpha glycosidic linkage, it is always trans in respect to the 6th carbon. You see here how the bonds are linked downwards in respect to that upward 6th carbon. But now, for the formation of beta glycosidic linkages, we have beta glucose pairing up with another beta glucose through the same condensation or dehydration reaction. So what is a condensation reaction? Again, it's the removal of a hydroxyl group from one of the rings and a hydrogen groups from the other ring. Now, when we remove a hydroxyl group and a hydrogen group, it will produce as well H2O outside the ring. This will form the beta glycosidic linkage that is pointing upwards, cis, in respect to that 6th carbon. You see here how it's pointing upwards to this 6th carbon. Thank you, Carbo Fitness, for having me. Take care. Now that we know what carbohydrates are, we can look at how they function. The basic biochemical roles of carbohydrates are to provide and store energy, as well as provide structural support. Carbohydrates are used to store energy since they contain reduced carbons, CH, bonds. Carbons hydrogen bonds have high potential energy because the electrons are shared equally by atoms with low electric negativity. Due to its hydrophilic property, carbohydrates digest quickly, thus providing energy quickly and efficient.
Therefore, carbohydrates are effective energy molecules since they store a lot of energy in their carbon-hydrogen bonds and because they are efficient in providing energy as well. In animals, the most common molecule for energy storage is glycogen, while plants, on the other hand, make carbohydrates through photosynthesis and store the energy as starch.
Although carbohydrates are primarily used for energy and structural purposes, they can be modified to serve other roles such as cell identity. This is done by covalently bonding different carbohydrate groups to a protein in order to form a glycoprotein that serves to display a unique identity for each cell. Since carbohydrates are such a good source of energy, most athletes use a method called carb loading, in which they consume an excess amount of carbohydrates before competitions so that they can perform at their maximum potential. And that was your daily dose of Carbo Fitness. Thanks for everybody who watched this episode of Carbo Fitness. I hope it was entertaining. See you next time.
To see more videos like this, visit the link below.